Let's discuss on translation process. Translation is a process which happens in all the living beings, including both prokaryotes and eukaryotes. And in this particular process, the protein synthesis happens over the ribosomes and using the RNA molecule as a template. So during the process of translation, the messenger RNA, which itself consists of 4 nucleotides, is translated into the proteins. So let's understand about the transfer RNA. What is a transfer RNA? It is consists of 4 arms. It is basically a clover leaf-like structure. The secondary folded structure of transfer RNA has 3 hairpin loops which give it an appearance of 3-leaved clover. And the main constituent of this transfer RNA are these 4 arms: the acceptor arm, the DHU arm, anticodon arm, and pseudouridine arm. So anticodon, the acceptor arm, that consists of 7 to 9 nucleotides of 5 terminal and 3 terminal. The 5 terminal has the phosphate group, and the 3 terminal has a specific sequence of CCA or CCA tail, and the amino acid attaches to 3 hydroxyl group of this acceptor arm via the phosphate group. So amino acylation of transfer RNA, or the charging of transfer RNA, is the first step in the translation process. And the enzyme amino acyl transfer RNA synthetase catalyze this particular reaction. Here there will be loading of amino acid. And the second arm is known as the DHU arm, and the D arm has a stem of 3 to 4 base pair, and it ends in a loop, that is the D loop. It generally contains the dihydrouridine. It is a modified nucleotides, and because of presence of this, it is known as DHU arm. Right. The next is the anticodon arm. It has the 5 base pair stem. It is an anticodon loop and present onto the messenger RNA for amino acid it carries. And these unpaired bases of anticodons, they loop anticodon loop pair with the messenger RNA codon, and each codon is identified by a specific transfer RNA. The next loop is the pseudouridine arm. And the T arm consists of a stem of 4 to 5 base pairs and a loop containing the pseudouridine. This is the pseudouridine base, and because of which it is named. And the next arm is a small short arm that is known as the variable loop, and it is present between the pseudouridine and the anticodon loop. In between these two loops it is present, and its size varies from 3 to 21 bases, and it helps in recognition of the transfer RNA molecule. So this is the overall structure and functions of various arms of transfer RNA. Then silent features of genetic code. So because genetic codes are present onto the messenger RNA, and the genetic information is right from DNA is transcribed in the form of these genetic code. So, and these genetic codes are decoded over the ribosome during the process of translation in the form of proteins. So what are the important feature of this genetic code? So let's have a look. It is a triplet codon. It is universal, degenerate, ambiguous, non-overlapping, and non-punctuated. So it is triplet because codes are onto the messenger RNA, and each codon is a consecutive sequence of 3 bases on messenger RNA, like U, O, U, U, and it codes for a specific amino acid. Similarly, AUG for methionine. And second important feature is non-overlapping. The codes are consecutive, and therefore the starting point is extremely important. The codes are read one after another in a continuous manner. So, and the non-punctuated, third important property is non-punctuated. So there is no punctuation between the codons. It is a consecutive or continuous chain of the codons. Then degenerate, because 61, there are 61 codons, but they code for 20 amino acids only. That means the one amino acid more than has more than one codon, and that's known as degeneracy, because serine has 6 codons, glycine has 4 codons, so it is the degeneracy of codes. Then wobble position in the codon and anticodon interaction, like CAGU, nucleotides of codon, then GUCA, usual nucleotide of anticodons, and isoleucine, UG, wobble nucleotide of anticodons. Then next feature is unambiguous, means without any doubtful meaning. The one codon stands only for one amino acid, and there are 61 codes but 20 amino acid. So next property is universal. Codons are same for the same amino acid in all the species. Then translation, move on to the translation process. It is the cytoplasmic process. The messenger RNA synthesized in the nucleus. And now it is coming to the cytoplasm, where the messenger RNA is translated from 5' to 3' end. It is read over the ribosome, and it is read in a manner from 5' to 3' end. And in this process, the polypeptide chain is synthesized, and the first amino acid is the amino terminal one, and the chain growth is from amino terminal to carboxyl terminal. The process of translation can be conveniently divided into the following 5 phases. So these are the 5 phases: activation of amino acid, initiation of the process, then elongation of the process, then termination. Then after that, post-translational processing of the newly synthesized protein. So let's discuss steps of translations one by one. The first is activation of amino acids. So activation of amino acid happens with the help of ATP and amino acid in the presence of transfer RNA, for which the anticodons are present onto the anticodon arm of the transfer RNA. And then ATP and amino acid transfer RNA synthetase is the enzyme. In the presence of this enzyme, the amino acid gets activated in the form of amino acid transfer RNA and AMP. So the two high energy bonds are being broken up, and that is equal to the investment of 2 ATPs. So in this reaction, ATP is hydrolyzed to AMP, and two high energy phosphate bonds are broken. So let's see the various steps of initiation. So before understanding the initiation process, ribosomal RNA, let's talk on ribosomal RNA. Ribosomes provide necessary infrastructure for which the messenger RNA and transfer RNA and amino acids, they interact with each other for translation process to happen. And this assembly is the protein synthesizing machinery. And nucleolus is the area where the ribosomal RNA is synthesized and ribosomal assembly is produced. Right? So initiation process, it does consist of various small steps, like recognition. First is the recognition of the site onto the messenger RNA from where it is to be started. And then formation of pre-initiation complex, that is the binding of tertiary complex of methionine transfer RNA and GTP and elongation factor 2. Then and the formation of 40S ribosome, 40S fraction of ribosome. Then binding of messenger RNA to PIC to form the 40S. This is the 43S pre-initiation complex. And then finally the 43S PIC. It combines with the 60S portion of the ribosome subunit to form the complete ATS assembly. That is the ATS ribosome. So what is happening here? So in the first step, the where the recognition step starts. So in eukaryotes, the first step the amino acid is incorporated is methionine, and the code present for this is the AUG onto the messenger RNA. But in the prokaryotes, the same codon codes for N-formyl methionine. That is the difference. And which is the first amino acid? The first AUG triplet codon after the marker sequence is identified by the ribosome as an start codon, right? And this upstream sequence in mammalian, this marker sequence is known as Kozak, Kozak, Kozak sequence. It will be somewhere upstream here. And in the bacteria and the prokaryotes, and the same sequence is known as Shine-Dalgarno sequence. As you have seen in ORI region during the replication, there are some marker sequences. Also in the transcription, there are some marker sequences are available. Similarly, for the translation to start, so before the AUG, that is the initiation codon, there are few sequences, marker sequences, which sense the ribosome to recognize this particular portion. And from that onward, there will be AUG, and the translation will be started from this particular site. So let's see here. This is the methionine transfer RNA. This is the activated amino acid with transfer RNA. Plus 1 GTP is being required, and elongation factor 2 is being required here. And at the same time, the 40S ribosome and elongation factor 3 and elongation factor 1, all these combine up and they lead to the formation of 43S pre-initiation complex, on which there is further loading of messenger RNA, leading to formation of 43S pre-initiation complex. And this process requires the investment of 1 ATP. So, and this total association is the 43S pre-initiation complex. Further, the initiation factor 5, initiation factor 2, and 60S ribosomal unit, all of them they combine and there is a formation of a complete pre-initiation complex, that is the ATS ribosome. And here also there is investment of 1 GTP, so GTP converted into GTP. So this ATS ribosome, the 60S component has two major sites. One is the P site, or the peptidal site, that carries the peptidal transfer RNA, and the second site is the what we call it is the A site, and that is the amino acyl site. So it carries the new incoming transfer RNA with the amino acid to be added next. So, and there is another site which lies onto the back side, it's not visible, and that site is known as E site. So after performing the function attachment of amino acid through peptidal bond, the empty tRNAs are knocked out by the exit site. So elongation phase. In the elongation phase, what is happening? You can see the attachment of new amino acid to the A site. So on the P side already, the very first amino acid along with the consent transfer RNA, on which the anticodon sequences are available, codon is available, that is UAC, against the AUG codon of the messenger RNA. So it is fixed onto the P side. The incoming amino acid along with the transfer RNA, specific transfer RNA containing the specific codon, anticodons, against the codons of the messenger RNA. So it attaches at this particular A site. Further, in the presence of elongation factor 1, an investment of 1 GTP is there, and what happens? This is going to form a peptide bond. So peptide bond formation happens between the methionine and threonine. Just as an example, methionine will always be the first one in the mammals and formylmethionine in the prokaryotes. And that will be the first amino acid. So first amino acid and the next amino acid in between, there is a formation of peptidal bond. There is no energy investment for the peptide bond formation because the amino acid is already in the activated form. So the peptidal bond is formed. Now there is a time for translocation, means inputting of these sites, shifting of A site tRNA to the P site, and P site amino acid has to donate the this methionine because it is now attached with the peptide bond, and empty tRNA now goes onto the back side on the exit side, and from there it is removed. So this UAC containing anticodon, this tRNA, you can see it here, it is knocked out via exit site. And this yellow light yellow portion, that is the P site, now is being taken up by the this tRNA, which contains the threonine amino acid. So it shifted it here. It cannot be, it not necessarily it is threonine, it can be any amino acid depending upon the type of codon present onto the messenger RNA. Then now A site is empty. And here in this translocation process, again, there is an investment of 1 ATP in the form of 1 GTP because there is a shifting. Now A site is being occupied by another transfer RNA containing the amino acid, activated amino acid. So you can see it here. Now this time glycine has come. Glycine along with the tRNA come. And then there is a further peptidal bond formation in between the threonine and glycine. And this is how the process goes and goes on. And the chain grows, the peptidal chain grows up. Then simultaneous reading and shifting of this complex, and the complex happens. And this is how the messenger RNA codons are read by this association of ribosome along with the tRNAs. So the translocation process, as I have already told, the tRNA fixed at P site does not carry any amino acid and is therefore released from the ribosome. Then the whole ribosome moves over the messenger RNA, and through the distance of one codon, the peptidal tRNA is translocated to the P side, and this is done with the help of elongation factor 2. Then A site is ready to receive another amino acid tRNA bearing the appropriate anticodon. The new amino acid tRNA is fixed to the A site by the base pairing with the messenger RNA codon. The translocation requires the hydrolysis of GTP to GDP. Right? This is all I have already explained in that previous diagram. Then sum up the energy requirement. What is the total requirement for this particular process of uploading of amino acid with tRNA and translocation? For each peptide bond formation, the 4 high energy phosphate bonds are used. Keep remember, the 2 for the initial activation of the amino acid in the form of like from ATP to direct AMP conversion, that is equivalent to investment of 2 ATPs, and then 1 for elongation factor 1, where the 1 GTP is lost to GDP, and 1 for EF2 step, where the GTP is lost to GDP. So in this particular process, there is a translocation of the amino acid and shifting of the ribosome over the messenger RNA. So actual bond formation does not require any energy because the amino acids are already activated. So the total you have seen, the 4 high energy phosphate bonds are being used in this process. Then post-translational modification. So it can be in the form of proteolytic cleavage. We'll see the example of proinsulin to insulin. Then modification of amino acids. Then it can be gamma carboxylation of glutamic acid. For example, in the prothrombin, which is prothrombin to thrombin, which is vitamin K dependent process. Then hydroxylation can be there. For example, proline and lysines in the formation of collagen, they get hydroxylated, and that is a vitamin C dependent process. You have already learned many a time. Then phosphorylation can happen, then glycosylation can happen, then methylation of stone proteins happens. Then finally, subunit aggregation. For example, you have seen hemoglobin. It is a uh, complex structure. So there are four subunits all joined with each other. Or another example can be immunoglobulin, then protein folding by chaperone. So these many processes happen in the course of post-translational modification. Few are reversible process and few processes are irreversible in post-translational modification. So like disulfide bridge formation, glycosylation, phosphorylation, acylation, N-acylation, acetylation, ADP, ribosylation, all these are reversible process. And while the proteolysis, ubiquitination, then lysine hydroxylation, proline hydroxylation, methylation, all these processes are irreversible. You can see the example of insulin. Right from proinsulin high, there is conversion, how there is a conversion to active or mature insulin. So this is the proinsulin structure, a long chain, polypeptide chain. So further conversion happen with the formation of inter and intra, intra chain, uh, inter or intra chain disulfide bond formation, followed by no splicing, cutting of these chains. Unwanted sequences are lost, and the two chains, A chain and B chains, are being formed, which are connected by disulfide bonds. Interchain and intrachain disulfidic bonds are there. So this is the structure of mature insulin right from the single polypeptide chain. So when the protein is being synthesized, it may assume different three-dimensional structure, out of which one will have only one will have the biological activity that you already know, and then abnormal folding of these protein may lead to the protein misfolding diseases. And chaperones are certain association protein molecules. They help to produce the correct spatial arrangement, and chaperones attach to nascent polypeptide chain and prevent the wrong folding. So they check any mistakes happening during the folding process of the protein, so that the folding is allowed only in one correct direction, and they help in assembly of tertiary and quaternary structure of protein. And chaperonopathies are the disorder resulting from sick chaperones, and these diseases progress with the age. Now here in this slide, we'll see the differences between the important differences between the prokaryotic and eukaryotic translation. Very, very important. Like with respect to ribosome first. So in the prokaryotes, 30S is the small subunit and 50S is the larger subunit associate to form the 70S. While in the eukaryote, it is 40S is small subunit and 60S large subunit. They together form the 80S ribosome. Then initiation, in the initiation process, the translation start before transcription is completed, and no messenger RNA processing or post-translational transcriptional modification happens in the RNA in the prokaryotes. Then in eukaryotes, the transcription and translations are well separated, and the processes and the processed messenger RNA is translated. Then consensus sequences. The initiating codon AUG is recognized by the Shine-Dar-Garno sequence present onto the messenger RNA that happens in the prokaryotes, while in the eukaryotes, it is the Kozak sequences which are being recognized by the ribosome. Then initiation of a ribosomal transfer RNA. So the initiating transfer RNA carries the formylated methionine, while in the eukaryotes, the initiating transfer RNA simple carries the methionine only. Then formation of initiation complex. So in prokaryotes, the initiation factor and GTP hydrolysis are required for the binding of a small ribosomal subunit to messenger RNA. Then in eukaryotes, the several initiation factors and the GTP hydrolysis are required for the formation of initiation complex. Then peptide bond formation. So in the prokaryotes, the peptidal transferase ribozyme is in the 23S ribosomal RNA, while the peptidal transferase activity lies in the 28S ribosomal RNA of the large subunit. Then post-translational events. So in the prokaryotes, it is mainly the co-translational modification. Then in the eukaryotes, you have just seen mainly the post-translational modification and then followed by sorting, then export, and then localization. So it all happens in coordination with endoplasmic reticulum. Then inhibition. Antibiotics can inhibit the different stages of translation in prokaryotes, while in the eukaryotes, the toxin like diphtheria, ricin, furomycin, and cycloheximide, all these are the inhibitors of eukaryotic translations. Then inhibitors of the translation, they generally act only on the bacteria and are non-toxic to the human beings. And this is because the mammalian cells have 80S ribosome, while the bacteria have 70S ribosome. And then reversible inhibition in the bacteria. So these antibiotics are bacteriostatic. Like tetracycline is there. It inhibits the attachment of amino acid RNA to a site of ribosome. Then chloramphenicol, it inhibits the peptidal transferase activity of the bacteria. Then erythromycin and clindamycin prevent the translocation process. So various step, these antibiotics check the translation process of the bacteria. Then irreversible inhibition in the bacteria. And these antibiotics are bactericidal. And streptomycin binds to 30S subunit of the bacterial ribosome, and they cause misreading of the messenger RNA and totally inhibit the protein synthesis. Further, the inhibitors of protein synthesis in eukaryotes, like furomycin, is structurally similar to tyrosine transfer RNA and gets attached to the A site of the ribosome. So the incomplete peptide is being released. Then cycloheximidine, it inhibits the peptidal transferase in 60S subunit, and diphtheria toxin liberated by the bacteria, Corynebacterium diphtheria, causes the inactivation of elongation factor 2 by attachment of ADP to effector elongation factor 2 and consequent inhibition of protein biosynthesis in mammalian system. Then inhibitors of transcription in this will also inhibit the